Hi, everyone. My name is Kevin Keegan. I'm the General Manager of Oncology at Illumina. I'm delighted to introduce Tom Stricker. Tom is the Senior Medical Director of Precision Medicine at One Oncology, based in Nashville, Tennessee. I'm really excited to have you here, Tom, and would you mind just helping us with a little broader description of what your role and how you operate within One Oncology?
Absolutely. So One Oncology is a management group that we have 14 practices that we provide a variety of sort of assistance to. And so for my role, we are trying to make precision medicine easier for the physicians so that we can more effectively deliver genomic medicine to our patients, and that we get the right test at the right time for the right patient, and we can get the right therapy based on those tests. So
that's fantastic. Really describing a precision medicine approach. I also am, keenly aware that you run a genomics lab within One Oncology. Can you talk about how the evolution in your field, and you have a background in pathology, and how the molecular profiling space and the genomic space in the context of cancer testing has come about?
Yeah, so it was for a long time. And even when I was training, we looked at slides and maybe we did some IHC and that was it. And then we discovered EGFR mutations in lung cancer. And pretty quickly after that, we had ALK fusions and ROS fusions and MET skipping. And so it became pretty clear that if you just sort of went down the list of so we're going to test for EGFR, and if that's negative, we'll test for KRAS, and if that's negative, then you'd run out of tissue, right? So you wouldn't, and it would take forever to get these. So pretty quickly it became clear we need panels, and then the panels got bigger, and then as sequencing improved, we were able to do even bigger panels, and that's even merged now into biomarkers that are a synthesis of large-scale sequencing, like tumor mutation burden, where it's not a specific mutation, but we're sort of looking at all the mutations you have and sort of counting them up and seeing how many there are. And so there's been just this explosion of genomic testing and precision oncology. And now we have to keep track of it all.
Great. So Tom, totally get it. I think all of these new biomarkers, gene signatures, you moderated a panel of experts yesterday, and the title of that whole forum was around ATGC spells hope. When you think about the technology, we've been talking about technology a lot at this forum, right? Translate that for us and for our viewers here about how that impact is creating hope for cancer patients.
Yeah, so there are I believe there are about 30 now FDA-approved biomarkers that lead to therapeutic decision-making. And the use of these drugs has a very significant impact on extending lifespan for patients. I used the example during our panel. There was a phase 3 lung cancer trial using standard chemo. It extended life 6 months, and this was hailed as a massive success. And the two curves both drop like this, and there's just a little bit between them. And now with drugs like EGFR inhibitors, if you have an EGFR mutation, or soratasib for KRAS G12C, we're seeing much more extended lifespan, more significant than 6 months, and that's just a big deal. So.
And those references are really focused on lung cancer.
Yep.
What about the rest? What other rare cancers or cancers beyond lung?
Yeah. So G12C that approval also carries for colon cancer. There's approval for that drug in colon cancer as well, and so KRAS G12C is mutated in lots of other cancers. We are seeing pan approval for some drugs. These tend to be for rare fusions, but if you have a NTRK fusion, regardless of tumor type, there's an approval for a drug to treat that. It's not common, but if you're one of those 1% that has it, it's a really big deal. And there have been absolute cures based on that drug. We see the same thing coming out with NRG1, where sort of a pan approval in that fusion. And so we're at that sort of cusp where we're moving from a little bit from tumor-based or the subtype-based sort of treatment to tumor-agnostic treatment, which is really exciting. So.
Yeah, that's a great thread. You run as a senior medical director in a lab that has a genomics lab. How is important to have or how much helpful is for your lab to have just a single workflow that can apply it across many or all cancers?
Yeah. For us, it's honestly, it's absolutely crucial. We are not big enough to have 30 different workflows. We don't have the volume to support all of that. It wouldn't make sense. And the way genomics works, it would honestly be silly to do. It just doesn't make any sense at all. I'm excited. We're talking about, especially with the new machine. What can we do on whole genomes that sort of let's really get down to one single test, right? Let's get down to the only test you need. So.
Yeah, since you brought it up, so let's talk about the genome, right? We use this term here at the forum that the genome era is here, is now. When you think about moving even from large panels of genomic profiling tests like comprehensive genomic profiling and moving toward whole genomes, what are the key enablers?
One of them's practical, so I hate to be very practical about it, but a technical validation of a genome is expensive because there's a lot of data, and so to prove that the assay works, it just requires running a lot of genomes, and it's going to take a little while. The other is getting payers comfortable with the idea. So, I think there's an idea that we can replace a lot of tests more cost and be cost effective than with a whole genome, but we probably have to go out and prove it to them, and that's fine. We can do that. We all believe. Everyone here believes in the genome, so we'll go prove them that this works. From a more sort of interesting point of view, are there data that you can get out of a genome that is tangential but useful, right? So, say we're doing a genome sequencing for a specific kind of cancer. But we're going to get PGX as well, right? We're going to get pharmacogenomics. So can we go ahead and leverage that? And also, can we create essentially living data or living reports? We have VUSs. There's going to be a ton of VUSs that come out of a whole genome, right? So when those VUSs, or if those VUSs turn into actionable variants, do we have a way to update the medical record, update the patients, update the oncologist, so that they can, if it's right for them, the therapy can change. So
yeah, that's really interesting. Well, I want to switch gears just a little bit. You serve you referred it to fairly small, but it's actually a fairly large oncology community practice network, right? One of the questions I have for you, we talked a little bit here at this forum around democratization of testing or democratization of cancer care more generally. And also, our CEO made this comment. We want to eliminate barriers that are based on geography or income or race or things like that. How do you see tools like our technology and the advancement of genomic technology as a way to serve patient populations in very diverse settings and backgrounds?
Yeah, it's so that is super important, and being able to get sequencing to underrepresented populations is really important. And so some of our practices do have significant underrepresented populations. Making sure that they have the same access to having these genomic tests done is one of sort of the I guess sort of my job is to, because one of the things we're trying to do is make sure that everyone who should be getting comprehensive genomic profiling, every one of the patients who comes to an oncologist or a group gets it. And whether that's internally or we're sending to one of the big partners that you guys have machines at, as long as they get, the most important thing is they get the testing.
And how can you see partnerships between diagnostic entities like Illumina, pharmaceutical companies, many of those represented here, and organizations like yours. Like, how can we team up in a better way so that we can create better access and awareness around the utility of this type of technology?
Yeah, so I'm really excited about this. We have really solid longitudinal data on our patients through our EHR. And genomes are powerful, but genomes get way more powerful when you have longitudinal data, when you know diagnosis, you have vital status, how long they survived, what medicines they've been on, how long they were on each of them. So when you can leverage that longitudinal data at scale in a community a community setting that stretches, so our practices stretch from New York to LA. I think there's just a tremendous amount of untapped power there that I hope that you and I and some of these pharma folks are unable to unlock, because it's going to be a lot of fun.
Absolutely, I couldn't agree more. Well said. I got to say, the exercise here at the Illumina Genomics Forum, I think it's been enlightening, it's been inspirational. People like you and your leadership role in the genomics space for oncology is truly amazing. I can't thank you enough for your time and participation here.
Well, I really appreciate the invite, and I enjoyed moderating the panel, and yeah, it's been a great meeting. And I think in some ways it has been super exciting and invigorating, and in some ways one of the things I've enjoyed about it is a lot of the problems seem so sort of technical and solvable. It's not nearly as daunting as one might think, I guess.
Yeah, and I think you started to use the word aspirational. I think we're all kind of in this mode of aspiring to a greater state, and of course in the interest of better patient care, and you're at the tip of the spear for all that. So thank you a ton, Tom,
Likewise.
for your participation, and thanks for the interview.
Thanks.
Thank you so much.